that lipid plays an even more important role in the body than was previously believed. The biological functions of lipids are largely driven by their diversity. Structurally, lipids are classified into 8 categories, such as glycerophospholipids, sphingolipids, cholesterol, fatty acid, and so on. They are further divided into main classes, subclasses, and molecular species, and it is now estimated that more than 40,000 lipid molecular species exist in the human body. In addition, there is a compositional diversity, the relative ratio of different lipids, which is observed at multiple levels between tissues and cells within an organism, between different organelles, and even between membrane leaflets and subdomains. Therefore, it is crucial to understand how this structural and compositional diversity is generated and how lipid structures and their local compositions are translated into biological functions. Among the diverse types of lipids, we are particularly interested in the diversity of fatty acids. Fatty acids are carboxylic acids with long hydrocarbon chains and are categorized according to their carbon chain length as well as the number and position of double bonds. In particular, we focus on fatty acid chain length regulation by fatty acid elongases, as we have identified one of the mammalian fatty acid elongases. Elongation is a central step in the de novo synthesis of long chain and very long chain fatty acids, and chain length is a critical determinant of their function and metabolic fate. We identified L6, a member of the mammalian fatty acid elongase family, as a target of SRBP1. L6 is a microsomal enzyme that catalyzes the elongation of saturated and monounsaturated fatty acids with 12, 14, and 16 carbons into C18 fatty acids, such as stearic acid and buxenic acid. This enzyme plays an important role in the de novo synthesis of long chain and very long chain non-essential fatty acids, working in conjugation with fatty acid synthase, stearoyl-coated saturases, and other elongases such as L3, L7, and L1. As you know, obesity, defined as an increased body mass index resulting from abnormal or excessive fat accumulation, is a disease that impairs our health. Overweight and obesity are commonly associated with cardiac disorders, including cardiac hypertrophy, remodeling, and cardiomyopathy. Importantly, many studies have reported that even non-obese individuals with multiple components of metabolic syndromes, such as hypertension, hyperglycemia, and lipid abnormalities, have an increased incidence and mortality of cardiovascular disease. Obesity perturbs lipid metabolism across nearly all tissues, leading to ectopic and excessive lipid accumulation. Under these conditions, lipids can become toxic, a phenomenon termed lipotoxicity. By focusing on the intricate pathways that regulate lipid metabolism, there is a potential to develop therapeutic interventions that improve cardiovascular outcomes in patients with cardiovascular disease. To investigate the role of L6 in vivo and its contribution contribution to lipotoxicity, we generated L6 knockout mice. As expected, L6 KO mice had significantly reduced level of C18 fatty acids and accumulated C16 fatty acids. When fed a high-fat, high-sucrose diet, L6 KO mice became obese and developed fatty liver to similar extent as wild-type mice. However, they were protected against diet-induced hyperinsulinemia and insulin resistance. Mechanistically, the improvement in insulin resistance observed in L6 scale mice was associated with altered hepatic fatty acid composition, restoration of AKT phosphorylation, and suppression of PKC epsilon activation. These findings indicate that hepatic fatty acid composition is a novel determinant of insulin sensitivity, independent of cellular lipid content. Taken together, our results suggest that therapeutic strategies of insulin resistance and type 2 diabetes should focus not only on the quantity of lipid, but also their quality. So far, we have analyzed the physiological and pathophysiological roles of L6 in various tissues and organs, and the results are summarized on this slide. The critical role of L6 in regulating fatty acid quality under lipotoxic conditions extends to multiple diseases. L6 deficiency ameliorates metabolic dysfunction-associated steatohepatitis, MASH, atherosclerosis, and heart failure. In addition, increased de novo lipogenesis is an early and common feature of cancer development, and we found that inhibition of L6 suppresses the proliferation of various cancer cell types, including breast cancer, bladder cancer, leukemia, and glioblastoma. On the other hand, L6 is also essential for maintaining normal physiological functions in the respiratory system, cartilage, and brain. Taken together, these findings suggest that L6, through its regulation of tissue and cellular fatty acid composition, influences diverse biological functions and contributes to a wide range of pathologies. The world of L6 has expanded far beyond my initial expectations. Currently, these projects are ongoing in our laboratory. Today, I would like to highlight two of them: muscle D MASH and lysosomal diseases and sphingolipidosis, and introduce them in more detail. First, let me tell you about our muscle D and MASH project. Metabolic dysfunction-associated steatotic liver disease, muscle D, encompasses a spectrum of liver disorders ranging from simple steatosis to MASH and cirrhosis, and is recognized as a hepatic manifestation of metabolic syndrome. A subset of patients with muscle D develop a progressive inflammatory subtype termed metabolic dysfunction-associated steatohepatitis, MASH, which can progress to end-stage liver disease. Since lifestyle modification and currently available pharmacotherapies for muscle D and MASH have only limited efficacy, there is a critical need for novel mechanism-based therapeutic targets. Because muscle D MASH is characterized by excessive lipid accumulation in the liver, alterations in lipid metabolism are thought to mentioned earlier, global L6KO mice were protected against diet-induced insulin resistance through alterations in hepatic fatty acid composition. To further define the role of L6 in the liver, we generated liver-specific L6 knockout mice. We observed that liver-specific KO mice, LKO mice, displayed significantly higher insulin sensitivity compared with Fox controls when fed a high sucrose diet, which activates hepatic fatty acid synthesis. Transcriptome analysis reveals that PNPLA3, whose protein product exhibited lipase activity towards triglyceride in hepatocyte and retinyl ester in hepatic stellate cell, was significantly downregulated in LKO mice. Moreover, adenoviral restoration of PNPLA3 reversed the enhanced insulin sensitivity in high sucrose diet-fed LKO mice. Although the data are not shown here in details, our findings demonstrate that liver-specific deletion of L6 enhances insulin sensitivity in mice by reducing the generation of stearic acid-containing ceramide, which mediate endogenous activation of protein phosphatase 2A, a negative regulator of insulin signaling. Collectively, our observations establish a functional link between L6 and ceramide metabolism, providing a new insight into the molecular mechanism of insulin resistance. In this study, we also examined the effect of liver-specific L6 deletion in leptin-deficient OBOB mice, a well-established model of obesity, insulin resistance, and muscle D. The OBOB LKO mice were obese and developed fatty liver. However, they exhibited lower plasma glucose and insulin levels and less severe insulin resistance compared with OBOB mice. These changes were associated with marked alterations in hepatic fatty acid composition, including an increase in C16 fatty acids, a decrease in C18 fatty acids, and elevated proportion of linoleic acid and DHA. High-fat diets are often used to induce muscle D, but in such cases, it is difficult to determine whether changes in hepatic lipids contribute to the pathogenesis of muscle D or simply reflect differences in dietary composition. Because OBOB mice developed muscle D even when fed a normal chow diet, we consider these models particularly suitable for evaluating changes in lipid profile in muscle D and for assessing the impact of hepatocyte L6 deficiency on disease progression. To investigate the impact of L6 deletion on muscle D, we performed untargeted lipidomic analysis of liver samples from 4 genotypes of mouse. Principal component analysis of lipid metabolites revealed clear distinctions among the FLOX, LKO, OBOB, and OBOB LKO groups. The Volcano plot indicates a significant increase in 106 lipid species and a significant decrease in 119 lipid species in the livers of OBOB LKO mice compared with OBOB mice. This slide shows heat maps of the top 30 lipid molecular species that were either increased or decreased in the livers of OBOBLKO mice compared with OBOB mice. Many of the downregulated lipid species contain C18 fatty acids, consistent with our findings that LB6 deficiency reduces C18 fatty acids. In contrast, all the upregulated lipid species were phospholipids, including 11 cardiolipin species. Cardiolipin is a unique phospholipid found exclusively in the inner mitochondrial membrane. Cardiolipin has a distinctive structure consisting of 2 phosphate residues and 4 fatty acid chains. It is essential for the organization of mitochondrial structures such as cristae and contact site, and plays critical roles in respiratory supercomplex assembly and function, mitochondrial dynamics, and mitochondrial protein biogenesis. Mitochondria are key organelles that play a vital role in energy production through glucose, glutamine, and lipid metabolism. Alterations in mitochondrial structure and function are recognized as a hallmark of muscle D. In muscle D, mitochondria are subjected to prolonged oxidative stress, increased fatty acid oxidation, and enhanced flux through the TCA cycle. These changes ultimately result in electron transport chain dysfunction, excessive production of reactive oxygen species, and mitochondrial damage. Next, we examine the livers of these mice by electron microscopy and observed significant differences in mitochondrial morphology. As shown here, compared with frogs and LKO mice, OBOB mice exhibited extremely large, swollen mitochondria with disorganized cristae, a phenomenon known as megamitochondria, which is frequently observed in muscle D and MASH. In contrast, OBOB-LKO mice displayed small, elongated mitochondria with well-developed cristae. To assess mitochondrial function, we measured mitochondrial respiration in primary hepatocyte isolated from the different mouse genotypes using a seahorse flux analyzer. Basal respiration, maximal respiration, and spare respiratory capacity were significantly reduced in OBOB hepatocyte compared with frogs and LKO hepatocyte. In contrast, OBOB-LKO hepatocyte shows preserved basal respiration, maximal respiration, and spare respiratory capacity relative to frogs and LKO hepatocyte. These findings indicate that hepatocyte LB6 deficiency improves mitochondrial function in the livers of OBOB mice. When focusing on cardiolipin, principal component analysis and heat map analysis revealed clear differences in cardiolipin profiles among frogs, LKO, OBOB, and OBOB-LKO mice. Furthermore, the heat map shows that the cardiolipin profile in OBOB-LKO mice did not return to normal, but instead was maintained in a distinct allostatic state. This slide shows heat maps of the total chain length and total double bond number of cardiolipin in the livers of frogs, LKO, OBOB, and OBOB-LKO mice. Focusing on chain length and double bond number, the total carbon number higher in the livers of OBOB mice compared with those of frogs and LKO mice. This indicates an increase in cardiolipin species containing very long-chain polyunsaturated fatty acids in the livers of OBOB mice. In contrast, hepatocyte-specific L6 deletion reduced both total chain length and total double bond number of cardiolipin species in the livers of OBOB mice. We have demonstrated that hepatocyte L6 deficiency alters cardiolipin fatty acid profile and improves mitochondrial morphology and function in the livers of OBOB mice. But how does L6 deficiency alter the cardiolipin profile? As shown here, nascent cardiolipin is synthesized by the conjugation of two phosphatidylglycerol molecules with non-specific fatty acid incorporation. It is then remodeled into polyunsaturated fatty acid containing cardiolipin, particularly linoleic acid containing cardiolipin, through the action of PNPLA8 and cardiolipin remodeling enzymes such as tafazine, monolysocardiolipin acetyltransferase 1, and ALCAT1. We analyzed the expression levels of genes involved in cardiolipin synthesis and remodeling in the livers of frogs, LKO, OBOB, and OBOB-LKO mice. As shown here, the expression levels of these genes were similarly elevated in both OBOB and OBOB-LKO livers. Among them, we focused on ALCAT1. ALCAT1 is a lysocardiolipin acetyltransferase that catalyzes the pathological remodeling of cardiolipin in response to oxidative stress associated with diabetes, obesity, muscle weak, and cardiomyopathy. This remodeling leads to increased loss production and mitochondrial dysfunction. It has been reported that ALCAT1 selectively increases the level of cardiolipin species containing very long-chain polyunsaturated fatty acids. Because cardiolipin is highly susceptible to oxidative damage due to its proximity to loss production site in the inner mitochondrial membrane, the incorporation of PUFAs may promote cardiolipin peroxidation and mitochondrial dysfunction in muscle D state. Therefore, we hypothesized that changes in fatty acid composition caused by L6 deficiency might suppress the pathological remodeling of cardiolipin mediated by ALCAT1. To test this hypothesis, we induced fatty liver in frogs and LKO mice by feeding them a high-fat GAN diet and used an adenoviral vector to overexpress either GFP or ALCAT1 specifically in the liver. On day 14 after adenoviral infection, we analyzed the fatty acid composition of cardiolipin in the liver using gas chromatography and mass spectrometry. As previously reported, ALCAT1 overexpression increased the proportion of polyunsaturated fatty acids in cardiolipin, particularly the homogamma-linoleic acid, arachidonic acid, and DHA. L6 deficiency tended to reduce the relative abundance of the homogamma-linoleic acid and DHA, and almost completely abolished the increase in arachidonic acid. Interestingly, ALCAT1 overexpression decreased the proportion of succinic acid, whereas LB6 deficiency reversed this reduction. To further investigate the impact of LB6 inhibition on pathological cardiolipin remodeling induced by LCAT1, we overexpressed LCAT1 and concurrently knocked down LB6 in the mouse hepatocyte cell line AML12 and assessed mitochondrial respiration using Seahorse flux analyzer. As expected, LCAT1 overexpression significantly reduced the oxygen consumption rate, reflecting impaired mitochondrial function. Notably, LB6 knockdown efficiently restored OCR level, counteracting the detrimental effects of LCAT1 overexpression on mitochondrial respiration. These results suggest that LB6 deficiency inhibits pathological remodeling of cardiolipin by LCAT1. In summary, these results demonstrate that LB6 regulates the elongation of C16 to C18 non-essential fatty acids and serves as a critical metabolic checkpoint. Hepatic LB6 deficiency improves insulin sensitivity independent of hepatic lipid accumulation. In OBOB mice, LB6 deletion improves cardiolipin composition, mitochondrial structure, and function in the liver. These improvements may result from reduced pathological cardiolipin remodeling mediated by LCAT1. We now aim to identify the lipid species that are altered by LB6 deficiency that suppress LCAT1-mediated remodeling and to elucidate their underlying mechanism of action. On the basis of our result, not only lipid quantity but also lipid quality and fatty acid composition contribute to the progression of muscle D and the pathology of MASH, with LB6 as a key player in this process. Notably, LB6 deletion has been shown to attenuate insulin resistance, mitochondrial dysfunction, hepatic inflammation, and fibrosis. Together, our findings suggest that LB6 represents an attractive therapeutic target for MASH. To examine the possibility that LB6 inhibition could serve as a potential therapeutic approaches for MASH, we established a novel MASH model by feeding mice a high-fat diet and administering both carbon tetrachloride and the liver X receptor agonist T09. Mice with this model exhibited key histological features of MASH, including hepatic steatosis, ballooning hepatocyte, Mallory bodies, lobular inflammation, and fibrosis. When this MASH model was applied to wild-type and LB6 deficient mice, LB6 deficiency did not ameliorate hepatic steatosis. However, serum markers of liver injury, including ALT and AST, were significantly lower in LB6 deficient mice compared with wild-type mice. These findings suggest that hepatic injury in MASH is attenuated by LB6 deficiency. Real-time PCR analysis of hepatic gene expression revealed marked upregulation of inflammation and fibrosis-related genes in the MASH model. However, this upregulation was significantly attenuated in LB6 deficient mice compared with wild-type mice. His finding indicates that LB6 deficiency ameliorates hepatic inflammation and fibrosis in MASH. Furthermore, to confirm whether liver fibrosis was suppressed by LB6 deficiency, we performed Mason trichrome staining and Western blot analysis of alpha-SMA, a marker of fibrosis. As shown here, the blue stained fibrotic areas were significantly reduced in LB6 deficient mice compared with wild-type mice induced with MASH. Moreover, while alpha-SMA protein levels were markedly increased in the livers of wild-type mice with MASH, this induction was significantly attenuated in LB6 deficient mice. This result further supports the conclusion that LB6 deficiency suppresses hepatic fibrosis in MASH. Based on these results, we are currently conducting further analysis using lipidomics and single-nucleus RNA sequencing of liver samples to elucidate the molecular mechanisms by which LB6 deficiency suppresses MASH pathologies. In addition, to explore therapeutic strategies for MASH through LB6 inhibition, we are developing nucleic acid therapies targeting LB6 and evaluating their efficacy in our MASH models. We have successfully obtained antisense oligonucleotides that robustly suppress LB6 expression in the liver and aim to advance these antisense oligonucleotides toward future clinical applications. Next, let me tell you about our project on LB6 and sphingolipidosis. Lipids are essential for the brain as they are a major component of brain tissues, comprising about 78% of the dry weight of the axonal myelin cells and 35 to 40% of the neuron-rich gray matter. Sphingolipids are a class of lipids containing a backbone of sphingoid bases. Because they possess both hydrophobic and hydrophilic properties, sphingolipids are essential for maintaining plasma membrane structure. In particular, sphingolipids are highly enriched in the nervous system, where they are pivotal constituent of plasma membranes and play critical roles in brain development and function. Furthermore, we found that inhibition of LB6 has a significant impact on overall sphingolipid metabolism in liver, brain, cancer cells, and stem cells. Based on this observation, we hypothesized that inhibition of LB6 could represent a potential therapeutic strategy for sphingolipidosis, a group of disorders caused by genetic defects in lysosomal enzymes responsible for degradation of sphingolipids. Sphingolipidosis are a subgroup of lysosomal storage diseases caused by an inherited defect in the breakdown of sphingolipids, which are essential components of cell membrane and signaling molecules. Sphingolipidosis is caused by mutations in genes encoding lysosomal enzymes required to degrade sphingolipids. When the enzyme is missing or defective, specific sphingolipids accumulate inside lysosomes, leading to cellular dysfunction, tissue damage, and progressive clinical symptoms. Although clinical manifestation of different lysosomal storage diseases vary greatly, over half of lysosomal storage diseases are associated with nervous system degeneration symptoms. L6 deficiency alters the balance of sphingolipids by changing the fatty acid chain length incorporation into them. This observation led us to consider whether inhibiting L6 could potentially serve as a therapeutic approaches of certain types of sphingolipidosis. Therefore, we first investigate the impact of L6 deficiency on Gaucher disease, a representative sphingolipidosis. Gaucher disease is the most common lysosomal storage disorder. Gaucher disease is a rare autosomal recessive genetic disease caused by mutation in the GBA1 gene. These mutations result in markedly reduced activity of the lysosomal enzyme beta-glucocerebrosidase, which hydrolyzes glucosylceramide into ceramide and glucose. Consequently, mutations in GBA1 lead to the pathological accumulation of glucosylceramide and glucosylsphingosine. Gaucher disease presents with a spectrum of clinical symptoms ranging from hepatosplenomegaly, hematological abnormalities, bone pain, and respiratory difficulties in type 1 Gaucher disease to severe neurodegeneration and premature death in type 2 and type 3 Gaucher disease. Type 1 is the most common form, whereas type 2 and type 3 are less frequent in many countries such as United States, Israel, and Brazil. However, higher proportions of patients with Gaucher disease types 2 and 3 are observed in parts of the Middle East and in Asian countries, including China and Japan. Type 1 Gaucher disease can be effectively treated with enzyme replacement therapy and substrate reduction therapy. However, these therapies are ineffective for type 2 and type 3 Gaucher disease. Therefore, the development of effective treatments for the neurological forms of Gaucher disease is urgently needed. This slide shows the fatty acid composition of glucosylceramide in human brain and the brains of a neuronopathic Gaucher disease mouse model. In both cases, the major molecular species in glucosylceramide containing stearic acid, a saturated fatty acid with 18 carbon atoms. Notably, stearic acid is the fatty acid synthesized by L6. In the brains of L6 deficient mice, glucosylceramide containing fatty acid with C18 or more were significantly reduced, whereas those containing C16 fatty acids were increased. To determine whether L6 deficiency suppresses the onset of neuropathic Gaucher disease, we conducted experiments using CBE, an inhibitor of GBA. In wild-type mice, CBE administration induced severe motor dysfunction associated with neuronal cell death and brain inflammation beginning on day 9, and all mice died by day 18. In contrast, Ergo-6 deficient mice treated with CBE showed no motor impairment and exhibited significantly prolonged survival. Wild type and Ergo-6 KO mice on day 14 after CBE administration. Wild type mice exhibited marked motor dysfunction, particularly affecting the hind limbs. In contrast, Ergo-6 KO mice displayed no motor dysfunction. They move freely within the cage and have grown to a larger body size. Here we propose potential mechanisms by which Ergo-6 deficiency improves the pathology of neuropathic Gaucher disease, as suggested by previous research findings, and how these approaches differed from existing substrate reduction therapies. Current substrate reduction therapy using glucosylceramide synthase inhibitors decreased all glucosylceramide species. In contrast, Ergo-6 inhibition selectively reduces the glucosylceramide species that accumulate in the brain of Gaucher disease patients and trigger inflammation. Furthermore, Ergo-6 inhibition is thought to markedly improve central nervous system symptoms by more effectively suppressing neuronal cell death and inflammation, while also altering the composition of other sphingolipids and phospholipids, and improving autophagy and mitochondrial function. Antisense oligonucleotides targeting Ergo-6 are currently under development as a potential therapeutic agents for this disease. Furthermore, GBA1 polymorphism represents the most significant genetic risk factor for Parkinson's disease. Approximately 5 to 50% of patients with Parkinson's disease carry mutations in the GBA1 gene, making it the most common genetic risk factor identified to date. Therefore, Ergo-6 inhibitors may also have therapeutic potential for Parkinson's disease in GBA1 mutation carriers, and research in this area is currently ongoing in our lab. This is the last slide. By using animal models, cell lines, and clinical samples, together with new research technologies such as lipidomics and single-cell RNA sequencing, we are working to identify and characterize lipids that play important roles in human health and to translate these findings into clinical applications. In parallel, we are aiming to determine the crystal structure of Ergo-6, elucidating the regulatory mechanism of its enzymatic activity, and apply this knowledge to the development of new Ergo-6 inhibitors. Moving forward, we will extend our investigation to develop new therapeutic approaches for lipid-related diseases such as metabolic syndrome, neurodegenerative disorders, and cancer, based on the regulation of fatty acid composition. I never imagined that our research would expand into such a wide range of directions. Thank you for watching our video. Please feel free to contact me if you have any questions or comments about our research. Thank you very much.